Today we are going to go over the basics of protein mutations. To start with, let's define mutations. A mutation is a change in the nucleotide base sequence. As you can see in this diagram, if a cell is building a protein, it starts with DNA that is transcribed into RNA and then translated into amino acids, which will create the protein. When RNA is translated into amino acids, it is grouped into 3 nucleotide bases, which is called a codon, as you can see in this other diagram. If an amino acid is incorrect, as it would be in a mutation, it can it can affect the structure and function of the protein because amino acids have different properties. So now let's go over the 4 different types of mutations. So at the top we have the original protein sequence, which is also known as the wild type sequence. The top line is the RNA sequence and the bottom line is the amino acid sequence. So our first type of mutation is a silent mutation. This is when one nucleotide base is changed, but it still codes for the amino acid. As we can see, the 5th codon over in the wild type is GGU, while in the silent sequence it is GGA. But if we look at our little diagram over to the right, it both codes for glycine. So there isn't a change in the amino acid, so the protein will not change either. The next type of mutation is a missense mutation. This is when one nucleotide base changes to code for a different amino acid. So again, in the 5th codon over, the wild type is GGU. But in the missense sequence, it is GAGU. We can see that the first one codes for glycine, while the missense codes for serine. This will change the protein's structure and function. Third, we have a nonsense mutation, which is when one nucleotide base changes and codes for one of the 3 stop codons. If we look up at the diagram, it says that UAA, UAG, and UGA are the 3 stop codons, which we can see here, and it has the little asterisks underneath. And the 2 after are just lines, because it stops the sequence, so the protein will be shortened, changing the function and structure. Lastly, we have a frame shift mutation. This is when a nucleotide base is added. As we can see, the highlighted blue A in the frame shift sequence is a new added nucleotide base, shifting all the other letters down. All the red amino acids have changed from the wild type, which will change the protein structure and function. You may be wondering why these mutations are important, and they are. They lead to diseases. For example, we have sickle cell anemia. If we look at the RNA sequence, we can see that in the middle codon, the A is changed to a U if you have sickle cell anemia, which changes the look of your blood cell. This is an example of a missense mutation. We also have cystic fibrosis, which is a nonsense mutation. As seen in red, we have a stop codon, which shortens the protein. So you can see in the picture that there is no protein channel. And lastly, we have Tay-Sachs disease, which is a frame shift mutation. We are adding 4 new nucleotide bases into the normal protein, which adds a new protein and then shifts the rest of them down, changing the overall protein. All right, and that is the basics of protein mutations.